When we transcribe DNA into RNA, we are not actually making mRNA. We are making what is called pre-mRNA. This is the immature mRNA strand, and we need to make it into a mature mRNA strand that is ready to leave the nucleus into the cytoplasm to be translated. In prokaryotes, however, we don't have this. We directly have mRNA formed from DNA. But in eukaryotes, we form pre-mRNA from transcription, and then we need to splice the pre-mRNA to form mRNA. So we previously said that DNA in eukaryotes is made up of sections, the exons that code for proteins and the introns that do not code for proteins. So we need to remove these introns from the pre-mRNA to have only exon sequences that code for proteins to have a mature mRNA strand. Because if we left the introns, this would prevent polypeptide synthesis. So the intron sequences are removed and the functional exons are then joined together. This process is called splicing. We splice pre-mRNA into mRNA.